Thank you. Buenos dias. Nice to see everyone this morning. This is very intimidating. It's kind of like a 24-second clock in basketball. I have to fire before it gets down to zero. Okay, so I'm going to be talking about the mechanisms of chronic allograft failure, that is to say transplantation. And we actually do a very good job in the acute setting. We have very good immunosuppression. We learned how to give that to patients. But what we have lacked is an understanding of the pathways by which solid organ allografts eventually fail due to allograft ischemia. So you start with a great big open vessel like that, and progressively over the course of 5 to 10 years, that vessel narrows to the point that it's just a little pinpoint. And if that happens in a renal transplant or a kidney, we simply give you a new one, provided we can find one. If it happens in a heart transplant, the patient dies. So that's really what is provoking this particular project that we are working on. So we are looking at the process by which this occurs, this narrowing progressively over the vessels. It will kill 50% of patients within about 10 years. And it is something that does not respond to typical immunosuppression. It's a big deal, even though transplantation is not a huge public health menace. It is something that we devote a lot of resources to. And if the patients die within a short period of time, then it's not fulfilling its promise. So we are looking at the process by which host smooth muscle cell precursors are recruited, and specifically by looking at the inflammation and the endothelial dance together that leads to this narrowing of the vessels, the vascular intimal hyperplasia. Now, although this is for a small number of patients who are getting solid organ transplants, the pathways, as it turns out, are also relevant to things that affect all of us. Particularly atherosclerosis. The same narrowing occurs by similar mechanisms in atherosclerotic disease, and it also is a big problem in stent restenosis. So it's not just about transplantation. Our model system, wow, very sensitive. So we do little heart transplants in mice, and we also transplant aortas, and that's usually our model. But as you'll see, one of the things on our wish list is to get models where we can model this many body problem in the Petri dish. And progressively we're doing microfluidic modeling. We have the kind of standard cytologic and molecular biology techniques. And what we've found so far is that there are certain mediators that are necessary and sufficient. We know where some of the host cell precursors are coming from, and we're trying to now track those down. We even know what drives those, and we are looking at the cellular interactions that are upstream. So the wish list, we would like to be able to find these cells as they circulate, be able to model the whole process outside of in vivo transplantation, and we would like to get high throughput screens so that we can look at multiple drugs. Jump shot, good.